All right, let's get started. Hello, one and all. Good morning, good day, good afternoon, good evening, wherever you are in the world. Thank you very much for joining us. My name is Rita Freischlad of Tumbleweed Workshops and Events. I will be moderating today's webinar on the open call for application from Biomedics and Ono Pharmaceutical on new strategies to engage neutrophils in solid tumors. Today's speakers are Christian Tidona from Biomedics, as well as Shingo Hota and Kazuhiko Takeda from Ono. They each have a short talk prepared, and we will have time for a Q&A session at the end. But before we get started, let's have a quick round of introductions. Christian, would you like to start us off?
Thank you very much, Rita. Welcome, everyone. My name is Christian Tidona. I'm the founder and managing director of the Biomedics Institute. It's a pleasure to be here, and you're going to learn a little more about myself in my presentation. Looking forward to spending the next hour with you.
Thank you. Shingo-san, would you like to go next?
Hello, nice to meet you. I'm Shingo Hota. I'm Associate Director of the Discovery Research Alliance team in Ono Pharma, Japan. Thank you very much for joining our webinars. Thank you.
Thank you. And Kazuhiko-san? You're muted.
Sorry, good morning and good to everyone. So my name is Kazuhiko Takeda. I'm a director of Discover Research Center of Oncology in Ono, Japan. Thank you very much to joining our webinar.
Thank you. Before we get started with the talks, just a bit of housekeeping. If you haven't used Zoom webinar before, you can take a look at your Zoom window and look for a box that says Q&A. Please submit all of your questions via the Q&A box and not via the chat. And you can read, like, and comment on other questions you see in the chat, so we know which questions are most interesting to all of you. I will post a few useful links in the chat momentarily, including the link to apply for the call. It's also highly recommended to register in the Biomedics career space, even if you're undecided, so you receive updates on this call for application as well as on future calls. And finally, as you can see, this webinar is being recorded, and we will send you the link to this webinar tomorrow, this time tomorrow. So now, without further ado, let's kick off the talks with Christian, who will tell us about Biomedics, the bootcamp, and life in Heidelberg.
Thank you so much, Rita. Let me quickly share my screen. Okay, here we go. Good. So, before we start, maybe a couple of words about myself so that you know who's talking to you. As I said, my name is Christian Tidona. My background is virology. I did my PhD at the University of Heidelberg, the beautiful city you see in my back. And but always, already during my studies at the university, I already had two hearts in my chest, one heart of a scientist and one heart of an entrepreneur. So I started my first company during my PhD, a diagnostics company called Multimetrics in Regensburg. Then I joined a cell therapy company in 2002, which was working on human liver cell transplants for children with urea cycle defects, so genetic diseases. In 2008, I started a stem cell institute called HiStem, Heidelberg Institute for Stem Cell Technology and Experimental Medicine with Andreas Trump, which is still up and running, an institute that is focused on cancer stem cell research. In 2009, I switched to regional development and managed a 80 million euro public-private research and development fund at a company called BioRN, bringing together academia and industry in Heidelberg. And in 2013, I decided to do something completely different. I started my own research institute, which is called Biomedics Institute. And I'm going to tell you in the next slides what it's all about. And during all this time, I was always following my passion. And my passion is actually helping early career research talents to grow. Now let's get back to the start of Biomedics. So roughly 10 years ago on the campus of the University of Heidelberg, today we have two sites, one institute in Heidelberg and one close to the campus of Yale University in New Haven, Connecticut. And in our institute, we're combining the best of both worlds, academia and industry. And we do this via a new, very unique innovation model, which combines global crowdsourcing with local incubation of the brightest ideas and talents. And we've actually published this model in 2015 in Nature Biotechnology. How does it work? It all starts with a very big question. We call it a call for application. And this call for application, we post worldwide at the best universities and research institutions and invite early career researchers to apply by submitting a very original project proposal to solve this particular challenge. And usually we get several hundred applications from 60, 70, 80 countries. And then jointly with our pharma partner, we select the 15 brightest individuals and we fly them in for a 5-day boot camp, in this case to Heidelberg, where they see each other for the first time. And we help them to associate in 5 competing groups and converting this collection of project ideas into something we would call a truly brilliant project proposal and a 15-minute pitch. And on the last day, they present their pitches in front of the jury, which includes the senior management of the sponsoring pharma company. In this case, it is Pharmaceutical from Japan. And if we agree on a winner, this winner is then relocated with his or her family to our institute in Heidelberg. We organize visa, jobs for their spouses, kindergarten spots, schools, new homes, whatever they need. And then they live and work for up to 5-year projects in our institute on the campus of the University of Heidelberg, where they implement their research project. This is what the atmosphere at such a bootcamp looks like. It's very energetic. This was a bootcamp we did with the German Merck on new strategies to enhance the immunogenicity of tumors. But what is very important at Biomedics, and this is also why we're doing these very intense bootcamps to select really the best of the best, is because we truly believe in our 3 core values. And if you visit Heidelberg, you will see them everywhere throughout our institute. The first one is cross-leverage. We inspire and motivate each other to unleash our full potential. What does it mean? Biomedics is a place for people who have embraced the concept of growing by helping others. So we want people to share their ideas, to help each other. And it's the exact opposite of some toxic atmospheres you would find in some of the top universities or companies where people work against each other. This is completely a complete no-go at Biomedics. We all help each other and we all support each other. And second is cross-pollinate. We embrace diversity as it is the source of innovation. So just imagine the more people that come from the more different backgrounds that are looking upon the same problem, the more likely it is that they will jointly come up with a truly innovative solution. And this is why we truly believe in diversity. Our research fellows come from around the world, from all different cultural backgrounds, religions, genders, science backgrounds, and it's good like this. We really embrace diversity, and we diversity because it's very important for the success of our institute. And third, aim high. Our goal is to enable a new generation of targeted therapies. We ourselves, including myself, we call ourselves the support team at Biomedics. So we support all the researchers so that they can be very successful. And we put a lot of effort in providing a really good research environment. But of course, we also expect that people make best possible use out of their limited period of time at the Biomedics Institute so that they grow as much as possible and also be as successful in their scientific endeavor as possible. We've been there, as I said, since about 10 years, and we've been working with 11 different pharmaceutical companies, including Ono Pharma. And with these companies together in the past 10 years, we have started 22 such research groups in Heidelberg, in our new site in New Haven, which just started about a year ago. We have already the first 2 projects. Then we also have a consortium. We are running a venture studio where the winner of the bootcamp actually doesn't become an employee of our institute, but becomes the founder and first employee of his or her own startup. And this is called Ion Labs in Israel. This is also Biomedics. And there we have started so far 6 different startup teams. We also publish. This is very important for our research fellows because most of the young researchers that are joining Biomedics, they're not yet decided whether they're industry or academic people. So it's very important, while you learn a lot about industry, that the way back to a tenure track and to a professorship is always possible. And this is why we always support also our scientists to publish well. And also we educate bachelor, master, PhD students And in Heidelberg and also now in New Haven, we have a very strong focus on deep disease biology and more artificial intelligence. In Heidelberg, there's 3 different areas in which we are active in almost all of our projects. The projects are dispersed across more or less all major therapeutic areas. But what is very important for us is to work a lot with human materials. A mouse is not a human, and it's very important that we really learn about deep human disease biology. And this is why quite a few of our teams are getting even whole human organs from multi-organ donors, and then they build from these primary cells very complex organotypic culture models where they learn about deep disease biology. And as you can imagine, with state-of-the-art technology, they're generating very deep multidimensional biological data sets. So almost all of the teams have dedicated bioinformaticians or are working with external bioinformatics and data scientists, companies and institutions. This is also very important for us. And sometimes a team is entirely focused on an AI solution, so purely dry lab. This is happening at Biomedics as well. And the third area is sensor development. We had a team that developed a diagnostic sensor, a printable diagnostic sensor based on graphene. And now, for example, we have a team that is developing fiber optic sensors with a completely novel sensing principle that can be implanted into the brain of a freely moving mouse to measure pharmacology for psychiatric diseases. In these diseases, unfortunately, there is no other way than animal models to really study things like depression or like schizophrenia. And you have to imagine that all these scientists that were selected via calls and boot camps, they're all encouraged to collaborate, they help each other, and they're coming from all these different fields, and they're all contributing to each other's science. And of course, this does not happen in the void. It's very important to be in a very vibrant research environment. And our first institute, Biomedics Institute in Heidelberg, of course, is in one of these major biomedical innovation hubs in Europe. On the left side, you see a map with about a driving radius of 1.5 hours around Heidelberg. And in this field, for example, we have 7 global pharma companies with research sites with 50,000 employees and 9 universities and research institutions, and we're collaborating with most of them. The centerpiece in Heidelberg is our campus in Neuenheimer Feld of the University of Heidelberg. It's more or less a square mile with more than 100 institutes, around 22,000 jobs in biotech and medicine, plus 16,000 students. And this is all on this square mile, including several startups, up close to 100 startups. And we operate in the technology park. We operate roughly 2,000 square meters office and lab space for our teams, right in the middle of the campus. These are some of the institutions you might know we're collaborating with in Heidelberg, and we have access to their core facilities. So whenever our teams need some very large piece of equipment or some or human samples or need to be in a very special lab environment, we're collaborating with the European Molecular Biology Laboratory, for example, the University of Heidelberg, and also the Cancer Research Center. Now, in addition to that, each of our teams starts several collaborations, and this depends very much on the focus of the project. So far, we have more than 60 collaborations worldwide with top research institutions. Altogether, we are operating 4 different sites. In addition to Heidelberg, I mentioned Ion Labs in Israel, next to the famous Weizmann Institute of Science in Rehovot. Then also close to Yale University in New Haven, Connecticut. And then also we have a very new model where we put Biomedics in a box on a pharma campus, and Boehringer Ingelheim is our first partner in Richfield, Connecticut. And all of these job opportunities, all these calls, everything is posted on our career space, which Rita already mentioned. Now let's go to the activities. So let's assume you're joining Biomedics. What can you expect? And we put a lot of effort into, as I said, into helping people to collaborate and to engage. And there is engagement on multiple levels. So for example, there is a bottom-up groups, we call them explore events and explore groups that are looking at career development and new technologies. There is a green team to avoid, to have a sustainable lab environment and to avoid plastic waste. Then in the field of science, every Friday, all the teams are meeting in one room. The group leaders decide who presents in front of everyone, and everyone contributes and helps. And this is usually a very good place to show a failed experiment and troubleshoot together with all your fellow scientists. And then we have an annual retreat where we come together for 2 days in an offsite and we all discuss science. And of course, we have a lot of fun with it as well. Education, I'm teaching entrepreneurship at the University of Heidelberg, and you can join the Scientist Entrepreneur course where you can learn how to turn a scientific finding into a startup pitch. We have scientific writing courses, German classes, but of course you don't have to learn German. Heidelberg is a very international place, and at Biomedics we speak English. There's the lunch talks, which is a format where each of our group leaders invites once per year a top scientist, a key opinion leader from around the world, and then we invite the entire campus in Heidelberg, and we do this in collaboration with the German Cancer Research Center postdoc network. We do sports together. There is charity runs, there's half marathons, there is dragon boat racing, which is pretty cool, climbing, yoga, and also a lot of social activities like potluck, where we bring foods from all our home countries, more than 25 countries at the moment, and we share. We have beer hours organized by the different teams. We have an annual summer party and a year-end cooking event. These are just some of the photos of our scientific retreat and the past dragon boat race, where, for example, our topic was the Smurfs. And the value, why should you come to Biomedics as research talents? Well, first of all, it's a fully funded 3 to 5 research group. You can fully focus on outstanding research and publications, so no teaching obligations. Creative cross-cultural teamwork atmosphere, guidance by experienced mentors from academia and industry. So every team is meeting once a month with the academic and industry mentors. You get introduced to a lot of senior managers from big pharma companies. It's an outstanding environment with direct access to leading research institutions locally, as you saw in Heidelberg, all within walking distance. You get a lot of leadership and entrepreneurship training. This is especially for the group leaders. And of course, all in all, it's a risk-free assessment of a possible future career in a startup or big company, because at Biomedics, as I told you, we publish. If you don't like the industry or if you don't like a company atmosphere, then you can always go back with your publication record and become a professor. Can we attract top talents from around the world? These are just some of the logos where people came from before they joined Biomedics, and you can see they're coming from all the top schools. So where are they going once they finish? About 50% go to industry, and the other 50% go back to academia. So before I hand over to Shingo-san and Kazuhiko-san for their presentations from Ono, I just would like to invite you to register at career.bio.mx in any case, even if this is not your preferred call at the moment. We are publishing more or less on a weekly basis new job opportunities and new calls for application on this platform at all our sites. And if you look at our website and LinkedIn and connect with us, you will find out that we are in the process of generating even more sites worldwide, which means more opportunities for you to join our environment and our ecosystem. So register at career.bio.mx or connect with us on LinkedIn. Also personally, if you want to connect with me, for example, just send me a quick note via LinkedIn that you participated in the webinar, and I'll be happy to accept your connection request. And with that, I give back to Rita.
Thank you very much, Christian. I see we already have questions pouring in, so that's great. Keep them coming. We'll get to your questions after the next talks. And with that being said, Kazuhiko-san and Shingo-san will be presenting now about Ono and a bit more in depth about the call topic. So whenever you guys are ready.
Yeah, thank you very much. So, yeah, hello everyone. From this point, Kazuhiko and I will introduce Ono and our call briefly. Just a moment. Can you see my slide? Okay.
Perfect.
Yes.
Okay. Yeah. Okay. So yeah. Thank you very much for joining this webinar. So we are also joining this live webinar from Japan, where it's currently 3 a.m. But please don't think that you must work until these hours of the morning or night, which are not always clear if you join our team. Okay. And before entering our main topics, so we would like to introduce ourselves quickly. So could you please introduce yourself, Kazuhiko-san?
Thank you very much for introduction me to. So my name is Kazuhiko Takeda. I'm a director of Research Center of Oncology Ono Pharmaceutical in Japan. So good evening or morning to all our attendees from around the world. I'm truly grateful to see so many of you here today, and we want to express our sincere appreciation for your interest. So my background is immunology and oncology. So I'm working on pharma more than 20 years. So I have a lot of experience in the oncology area. So firstly, my experience, most experience is immuno-oncology. So I was in charge of the nivolumab, as you may know, the Opdivo. So we are developing with the Medix and acquired in the BMS. So and then I moved to the Discovery Research Alliance, which team taking care of the collaboration with the academia and also the biotech company and then I moved to the Ono Pharma USA, a subsidiary in the United States. At that time, we have the subsidiary in the New Jersey. So I was working on Ono Pharma USA for 3 or 4 years, and then moved back to Japan. And I was associate director in the oncology research team, and also I lead some immuno-oncology projects. So one is EP4 antagonist and STING agonist. Right now, that's compound what is in clinical trial. Thank you very much.
Okay, thank you, Kazuhiko-san. So, okay, next I would also like to introduce myself. So, I'm Associate Director of Discovery Research Alliance in the Oncology Research Center. I have been with Ono since 2007, and that is about 18 years now. So I have got a background in cancer biology and electrophysiology, and actually I got my PhD in electrophysiology, focusing on the smooth muscles. But since joining Ono, I had been researching some kinase inhibitors. So predominantly, I have involved myself in high throughput screening, biomarker discovery, and IND-enabling studies for BTK inhibitor, Berexibel. So you might have heard of it. It's currently being used for the treating PCNSL and lymphoplasmacytic lymphomas. So in 2016, I moved to the Discovery Research Alliance team. And since then, I've been working for the collaboration opportunity with biotech and academia. In this time, I have moved to our subsidiaries in US and UK, spending about 2 years in each place. So while in the UK, I spent a year leading the Share UK Life Hack Alliance as the scientific lead. So after that, I came back to Japan in 2021, and since last October, I've been working as an associate director for current team in Japan. So, okay, and before we delve into our main topics, so I would like to offer a brief introduction on Japan and Osaka, as Ono's headquarters is located in Osaka, Japan. So Japan is a archipelago comprised of 4 main islands and over 6,800 smaller islands, so inhabited by a population exceeding 126 million. So the country is famous for the unique culture practices, so including tea ceremonies, flower arrangement, and calligraphies. So the country is also famous for its innovative technologies and is home to some of the biggest electronic and automotive companies in the world. So as you may know, it's Toyota. So, and as for Osaka, it's the third largest city in Japan, with a population exceeding 8.7 million people. So the city is famous for its vibrant atmosphere, bustling shopping district, and delicious street food. So here is a picture of takoyaki. So a popular popular Osaka dish. So taco translates to the octopus, and yaki means baking in Japanese. So takoyaki is a baked white dough filled with a minced octopus. So one of the Osaka's most distinguished landmark is Osaka Castle, a historical monument dating back to the 16th century. So the city also hosted Universal Studio Japan, well-known tourist attractions. So yes, that our personal location now introductions. So now I will hand over to Kazuhiko-san to provide the brief information about Ono.
Yeah, thank you very
Okay.
much. So from this slide, I'd like to introduce our company, Ono Pharmaceutical company. So as Singh-san mentioned previously mentioned, our headquarters is in Osaka, in Japan. So our revenue of last fiscal year is $3.4 billion US, and we have over 3,700 employees, which means that our company size is in upper middle range. So we have the very long history, more than 30 years. So we have the very long history, but Ono accelerated its modernized R&D activities after focusing on the prostaglandin research in 1965. So next slide, please. So that slide shows our financial trends. I would like to emphasize our R&D costs and the corresponding ratio. Our R&D expenditure lies over $730 million US, accounting for around 21% of our total revenue. That means we are focusing on drug discovery and development, aiming to create innovative drug like the cancer immunotherapy Opdivo. Next slide, please. So at present, our products are primarily sold in three therapeutic areas. One is cancer supportive care, and diabetes, and also immunology. That's the main area. And also that we are focusing on these three areas on the research side. So next slide, please. So that slide shows our domestic operation. So we have the two research institutes. One is Minase Institute between Kyoto and Osaka, and one is another one is the Tsukuba Research Institute, north part of the Tokyo. And also we have the instrumental in generating innovative drug, including I'm sorry. So we have our own manufacturing plant and the development team and the marketing and the sales branch in all over Japan. So next slide, please. So, and also that we have global operation. So we have 5 subsidiary in all over the world. So one is US side, and also one is UK side. So right now in UK and also the US side, we have the clinical development team. So we are now growing our capability in all over the world. So next slide, please. So this is our key slide. So we are focusing on the totally 4 specific areas. So one is oncology, and one is immunology, and neurology, and specialty. So we are very focusing on the open innovation. The open innovation means scouting the early stage compounds, the exploiting breakthrough modalities, and exploring the innovative seeds, and also combine to the technology, informatics, and model, and compound synthesis technology, and then we develop the unique and innovative new drugs. Next slide, please. So I'd like to explain our history of Opdivo. So as I mentioned, we are very focusing on the open innovation. So early 90s, we start collaborate with Kyoto University. As you may know, the Nobel Prize, Dr. Honjo, and then we find the PD-1 antibody. So, and then, so, but we don't have any expertise to develop the antibody. So we collaborate with Biomedics, and then acquired by the BMS, but still continue the relationship with BMS and develop the PD-1 antibody. So right now, the Opdivo is growing up, and approval more than 12 cancer type approval in the world. Next slide, please. So, and also that we have some new type of drug after approving the Opdivo. As you may know, the CTLA-4 antibody or proteasome inhibitor, Kyprolis, and a BRAF inhibitor or MEK inhibitor. Also the Shingo-san related in this project, BTK inhibitor, Berexibel. So we are expanding our oncology capability. So next slide, please. And as I mentioned, we are very focusing on the open innovation. So this slide shows our collaborator in the world. And we have a lot of collaborator in each research therapeutic area. So next slide. And also we have a lot of collaborator with technology-based companies. So these companies have a unique technology. So as you may know, the one is Schrödinger is computational chemistry, and also right now we are focusing on the AI drug discovery, Helix and Ictos. So next slide, please. But not only biotech company, but also we have a lot of collaborate with academia. So right now, the more than 300 partner in Japan, and also the US and the UK. So we are very focusing on the open innovation with academia and the biotech technology. So So we are very interested in this opportunity collaborate with the Biomedics. So thank you very much. So lastly, that slide showed our corporate philosophy. Our corporate philosophy is dedicated to the fight against disease and pain. So with collaborate with the academia and the biotech company. Thank you very much.
Yeah, thank you, Kazuhiko-san. So, from here, let me introduce our core new strategies to engage neutrophils in solid tumors. So, the generation of immunity against cancer is a cyclical process that can be self-propagating, leading to an accumulation of immune stimulator factors that, in principle, should enhance and broaden T cell responses. The cycle is also characterized by inhibitory factors, which serve immune regulatory feedback mechanisms that can hold the development or limit the immunity. So this cycle can be divided into 7 major steps, and starting with the release of antigen from the cancer cell and ending with the killing of cancer cells. So a number of cancer immunotherapies, therapeutics have been developed that target these T-cell-based cancer immunity cycles. For example, the anti-PD-1, PD-L1 antibodies modulate the process of cancer cell killing by T cells. And on the other hand, many clinical trials of immune checkpoint inhibitors and immune modulators with other therapies has been conducted, but many cancer patients remains resistant to these drugs. So, based on this circumstance, Ono is currently also focusing on the anti-tumor mechanisms that are independent of the T-cell-based cancer immunity cycle. So, which immune cells should we target for patients who exhibit resistance to drug targeting existing immune therapies? So, as you may be aware, subsequent to targeting T cells, many companies, including Ono, have been focusing on the macrophages. So, additionally, some companies are also concentrating on the NK cells. So, in the immune component in the tumor microenvironment, we are very interested in the neutrophils as the next target based on the following public information from the next page onwards. So regarding neutrophils, their function for cancer immunity remains inconclusive. Their association with cancer is evident and has been extensively analyzed in recent years. So, for example, neutrophils have been classified in greater detail, leading to clearer classification criteria. So in addition, the numerous studies have indicated that the presence of neutrophils is predictive of poor overall survival rates. On the other hand, progress has been made in the analysis of anti-tumor neutrophils recently, so suggesting that the higher ratio of anti-tumor neutrophils is linked to a better prognosis in pancreatic cancers. So, and this slide outlines potential strategy for targeting neutrophils. So there are many reports that mention the neutrophil possess dual pull and anti-tumor functions. For thinking of discovery of new drug utilizing neutrophils, so we might consider the strategy of activation of neutrophils or of neutrophil repolarization. So on the other hand, if we focus on the pro-tumor activities, the strategy of decreasing neutrophil, which has pro-tumor activity or blocking immune suppression function or immune suppression functions of the neutrophils might be considered. So, for example, the inhibition of trafficking and of netosis and so on. So, and in this slide, we summarize the agent being investigated in cancer and categorize these agents based on the strategies. So, some drugs related to the neutrophils regulations, for example, the PD-1, PD-L1 antibody related to the checkpoint engagement between neutrophils and T cells. So, in addition, the anti-IRAT antibodies has been developing in the clinical trial targeting neutrophil recruitment and other functions. So while some clinical trials of these agents that modulate the recruitment or phenotype of tumor-associated neutrophils in patients with solid tumor have been conducted, but there is still no clear evidence of effectiveness of these agents. So given the dual role neutrophil play in tumor microenvironment and the current situation of the developing agent, so we are eager to address the challenge of exploiting their activity and or modifying them to the anti-tumorigenic properties. So lastly, I would like to explain the Ono's core in more detail. So about this project, so we would like to focus on the novel strategies and potential target to harness neutrophil function in solid tumors. So based on the coming strategy, we will identify and shed light to the novel neutrophil mechanisms and biology. For validating the concept, so we also need to develop a robust in vitro assay system allowing the assessment of neutrophil functions. So in addition, we need to validate the finding target by using the generated assay system and some tool compound and antibodies. So on the other hand, we are not interested in the strategy of neutrophil depletion and of only suppressing the pro-tumoral neutrophils. So, and also we are not interested in the ideas on the device, and we are only focusing on the first-in-class strategy. We are not interested in the best-in-class strategy. So, and this slide outlines the type of researchers we are seeking. So ideally, we are looking for the researchers who hold a PhD or equivalent in immunology, oncology, or related field. So moreover, we require the researchers with strong knowledge and experience in the field of immunology, preferably neutrophil biology, and or oncology, coupled with the ability to propose innovative ideas. So additionally, it's important that applicant enjoys interacting with other researchers, including the members of the Ono team. And if you join our teams, Ono has potential to generate and provide tool antibodies to the teams based on your idea and validation data. So as a team member, you will gain insight into the strategies and thought processes of a pharmaceutical company and have the opportunity to interact with academic and pharmaceutical scientists from various fields. So in addition, the team will have the chance to visit our lab in Japan and experience the unique culture of Japan. Okay, yeah, that's it from us, and thank you for listening. And we are really looking forward to reviewing your innovative ideas and meeting you bootcamp. Thank you very much.
Excellent. Thank you very much. Thank you both very much for the great talk.